Hello, SYNGAP land. My name is Michael Graglia. Today is Monday, February 2nd. We are in week 6 of the year 2026. Before I jump in, there is a survey we want every single family in the U.S. to take. We want every single family in the U.S. to take. If you've been to ProMMiS, if you haven't been to ProMMiS, we want you to take this survey. It is in English and Spanish. Both links are the first 2 links in the show notes. Take the survey. Tell us about your experience at ProMMiS, or tell us that you haven't gone and why you haven't gone. We want to make ProMMiS the best possible experience for every family. We have sites in Philadelphia, Colorado, and Stanford. Go, go, go. But start today. Take the survey. It will take 1 minute. 1 minute of your time. First 2 links in the show notes. All right. Catherine Heldy, our CSO, who's just exceptional, has posted a blog about our funding priorities for 2026. I saw one family member write on Facebook. I read this. I have no idea what it means. That's kind of okay. You should understand the high-level thoughts, but really this is targeted at our grantees and young investigators out there to say, this is the stuff we're looking for. We're sending a signal. It takes them time and energy to write these grant applications and to engage with us. We want them to know what we care about. What do we care about? We care about genetics. We want to understand the genetics of SYNGAP1. As I'm about to tell you with another paper, not every patient gets diagnosed, right? So we want to understand the genetics of SYNGAP1. We want to understand where the other patients who aren't diagnosed yet are. We want to find them. Behaviors. Behaviors are the things that bring our families to their knees. They've brought me to my knees before. We have to better understand why the Syngapians have such extreme behaviors and how to help them. This is an area that has been understudied. We want to get it better. And isoforms. This gets a little geeky, but the SYNGAP1 gene is big and complicated, and it makes different sort of versions of itself. The way I think about it is like different lines. You know, I have a Subaru Ascent, but is it a touring model or whatever model? Is it white? Is it green? Do I have the fancy extras? Whatever. So that's how I think about isoforms. Same car, different makes, different sort of trim levels. All the scientists are like, oh, Mike, stop trying to explain science. But the point is, we need to understand those isoforms because different SYNGAP isoforms play different roles in the body at different times in life. And if we don't, and we need to understand that because we're dealing with these Syngapians for their whole lives, and we need to understand what is haploinsufficient and what is not. So excellent blog by Dr. Heldy. Read it, share it. If you're a scientist or a grantee, make sure you study it. Pubmed, by the way, speaking of publications, is at 6 for the year. So we're at 1 a week, which is a bit under where we need to be, but I'll take it. The coolest paper on Pubmed I read talked about a Syngapian who looked at 3 or 4 different diseases, but there's a SYNGAP case study in there. And basically this Syngapian was tested, but because they were in a GC-rich area, so remember DNA is in all these different letters, and some of these letters are G's and C's. And if you get an area that's got a lot of G's and C's, it turns out genetic testing 10 years ago, short read, didn't do a great job with that. And when they retested this patient with the long read, I'm not going to explain short read and long read because I'll butcher it, they actually got a diagnosis. So there's a lot of Syngapians out there who, just because of the quality of the genetic testing and the location of their mutation on which exon, it was missed. It was missed. That's an excellent paper, and it helps us answer that first question in Catherine's blog. Where are all the other patients? We know from different models, from talking to people, from looking at, from studying ourselves, that there must be more Syngapians out there. So why are we not finding them? Why are we not finding them? Well, one of the reasons is that part of that answer is which exons have GC-rich letters, and how dense is our population there? How many are we missing? So we're going to look into that. Again, we're so lucky we have a geneticist for our chief scientific officer because it all comes back to genetics. It also, though, if you've got a friend who you're like, this kid needs genetic testing, and the parents say something that rhymes with, oh, we got him tested a long time ago, they didn't find anything, you got to hit that person upside the head and say, what kind of testing was it? Was it chromosomal microarray? Because that's not going to do anything for a genetic issue. Or was it a year ago? Was it 10 years ago? Was it 50? How long ago was it? Because this stuff changes every year, guys. And what we're seeing now with long reads and some RNA work is that genetic testing right now is better than it ever has been. And if you are looking at another kid and you're like, that kid's life could be improved if the family knew what was going on, encourage that family to go after genetic testing. I just talked to a family last week. Kid's 17. They were tested 10 years ago. Didn't get anything. Got tested again. Boom. SYNGAP. So this is real, guys. We need to find every patient. I want to congratulate CURE SYNGAP1 Poland, and I want to ask formally, guys, I want one of those cool t-shirts. So CURE SYNGAP1 Poland, Basically, what we're trying to do is get the different SYNGAP groups who are spiritually aligned around working together, clinical trial readiness, investing in science, to just go with really simple names. We are CURE SYNGAP1, there's CURE SYNGAP1 LatAm, there's CURE SYNGAP1 Poland, we're starting other CURE SYNGAP1s, we have CURE SYNGAP1 Australia, because we want industry to know that we are all holding hands, singing Kumbaya, working together. We want clinical trials, we want them everywhere. So CURE SYNGAP1 Poland adopted that name, printed up t-shirts, and they hosted a meeting with our friends from the Camp 4 medical team to talk about Poland as a potential trial site. Huge work. Check out the LinkedIn post. Like it, comment on it. This is an example of a community of parents who will not take no for an answer and have time and again shown up on a level that I'm not used to seeing outside the US, except for the Argentines, because they're also a little crazy. And the Aussies are good too. But so really impressive work, CURE SYNGAP1 Poland. I hope you end up getting a clinical trial site out of that meeting. And I hope the rest of the world is watching and truly is impressed by what you're doing. Speaking of the rest of the world, a couple years ago, when we started getting excited about Stoke and Camp 4, I had a bunch of Europeans calling me being like, Mike, do I need to move to the US? What should I do? Blah, 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 blah. And I answered that. And now I've got Americans calling me, Mike, the Camp 4 is going to start in Europe. We have family in Europe. We're going to move to Europe, blah, 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 blah. Wherever you are, wherever you think clinical trials are going to be, don't move. Don't move. That's my answer to everybody. Couple reasons. First of all, it's a lot of work to move. And with a Syngapian and then to establish care, don't do it. Second of all, I am not in charge of who gets into a clinical trial. Camp 4 is not in charge of who gets into a clinical trial. Camp 4 contracts with clinical sites. Clinical sites are led by very senior clinicians who make decisions, right? They screen the kids. They say, yes, this kid should get in. The clinician at the site puts the kids in the study at the site. So if you're moving from country A to country B, you've got to believe that you're going to rock up in country B and walk into the clinician's office, if you can even get that far. And then you're going to say, look at my cute baby. They're so cute. Can I please get into the study? And by the way, I don't want to be on placebo, wink, wink. And they're going to be like, who the heck are you? Here's my 10, 5, 15, 20 SYNGAP patients who I already know and love that are from this country. Why are you here? That's what I said to the Europeans 2 years ago. That's what I said to the Americans last week. Do not move. If you want a clinical trial site in your country, check out what the Polish did. Check out what the Argentinians did. Organize your local patient community, wave your flag, work closely with SYNGAP1, get in front of our industry partners and explain to them why they would be crazy not to have a clinical trial in your country. That's the answer, team. That's the answer. Organize, advocate, bring the trials to you. So please stop calling me about moving. Don't move. Biorepository. We have a biorepository here in the US. We need more samples. There's a lot of ongoing studies. I don't have time to explain them all, but we need SYNGAP patients to be in that biorepository. We need your blood. We need you to consent. We need your genetic report, all those things. We have all these systems and processes. People will call you, but you've got to sign up. Mike, how do I do it? Where do I do it? Do I have to come to the conference in Denver? Yes, you can do it at the conference in Denver, but there's a lot of conferences between now and Denver, and our partners at Combined Brain, IRB approved, doctors, pharmacists, PhDs, these people are amazing. The chief scientific officer, by the way, over at Combined Brain is one of ours, a SYNGAP mom named Dr. Lindsay Wyzorek, who's just amazing. But there are conferences that are part of the Combined Brain where you can give blood for our biosample, and it will be collected by trained phlebotomists who work with kids who are very, very good. There's going to be in Texas, Florida, Maryland, Colorado, North Carolina, Massachusetts, whatever MI stand, Michigan, NC, PA, MA, MD, TX, PA, MA. I know what these states are, but it's just faster to read the letters. So guys, please, if you're in any of those states I just named, or there's a link in the show notes, look at the map. There's dates and places that you could take your Syngapian and donate to our biorepository. Super important. Last thing on my notes here is we've updated Champions of Hope. Every year we update big donors, 5,000 and up over their lives, and we list them. I want to thank those people from the bottom of my heart. We could not do this without our donors. I'm so grateful. And then there's two events. There's one event coming up I want to announce. On May 28th here in San Francisco, we are going to have a gala in San Francisco for CURE SYNGAP1. Ash and I are hosting. If you can make it, please join us. Thank you so much. Look forward to another pod soon. Things are happening, guys. The SYNGAP land is going fast.